First, thank you for the organizers for inviting me, that also the, let's say, northeastern region of Slovenia is represented at the conference from a beautiful city of Maribor. We have there something that Ljubljana is missing, and that is lovely vineyards. So if you are fans of good wine, then our region is the favorite one to visit. Yeah. I come from the Laboratory of Physical Chemistry and Chemical Thermodynamics, so the talk will be a bit different, more computationally oriented, but I will try to be very brief on one case, and on the other side also relatively precise. Okay, what's the structure of my presentation? First, I will present molecular docking and inverse molecular docking, actually invented by us. Then we will talk a little bit about drug repurposing, why it is important, especially in the case of rare diseases, and finish up with inverse docking for, of course, drug repurposing and inverse docking fingerprints. So I'm not sure how familiar you are with the process of molecular docking. It fits in the high throughput virtual scenario where you reduce the number of candidate molecules, candidate ligands for possible inhibitors and therefore drugs of some undesired protein target whose effect you want to block. So first, you need the structure of your protein receptor. You need to know its binding sites. And into those binding sites, you then dock like a library of possible candidate molecules. When I talk to my grandmother what is molecular docking, I say it's like hyperdimensional Tetris. Yeah, so the molecule must fit into the active site of the protein target by both, steric factors, just the shape complementarity, and of course the types of interactions. So what does molecular docking do? We start from really big in silico libraries of 11 million up to maybe even a billion of different molecules, and we want to find a suitable candidate molecule to inhibit our protein target. And molecular docking really nicely reduces in several steps this huge number of initial molecules to the so-called hit molecules, which later produce the lead molecule for subsequent pharmaceutical design. So, how does a normal docking work? We have the large protein target with its active site, and we have the ligand. And we want to fit the ligand both through sterical complementarity and different interactions into the binding pocket of the molecule. This is, of course, done entirely computationally. And we don't just get a single structure that is docked, but we get like an ensemble of docked structures here on the right side, where you have different poses of the same ligand within the same active site. And we can score these different poses by using different scoring functions. Most of them are energy-based, so how well does energetically fit our ligand to the protein target? Now, our question was, can we turn this procedure upside down? And the answer is, of course, yes. And we came up with a novel approach called inverse molecular docking. Here we have one molecule. Here I selected selamectin as a drug compound, and we want to dock this molecule into different protein targets to see to which of these protein targets our selamectin fits the best, both sterically and, of course, energetically. So, how does this work? One takes the ligand and then takes a library of protein targets. Here we usually use the entire protein data bank, which now has more than 300,000 entries for different protein structures. And what is the result? We find the protein structures, the protein targets that most likely bind our initial ligand molecule. And we are also able to rank these targets based on the energetic scoring function. Now, why would such inverse docking application be useful? Yeah, first, if we have a new molecule and we know its effects, but we don't know the molecular mechanism behind it, we can perform mode of action studies. So we can detect the likely proteins to which our molecule will bind. This is, of course, related to target fishing, but then there are two another important aspects. This allows us for selectivity studies, especially in the case of kinases. It's very difficult to inhibit only one kinase, but usually you inhibit several of them, but here we can predict how selectively our drug candidate will bind to a certain kinase. And another aspect of this is the off-target studies. So if you have a lead compound in your pharmaceutical pipeline, you know that it fits your protein target, it inhibits it, but you don't know what other possible targets it also can inhibit. And yeah, this can lead to, of course, adverse side effects. And you can already find this in front of the testing just by computational means. For our talk, the most important application is, of course, drug repurposing. So, what is drug repurposing? I mean, in plain, let's say, language, it would be the use of already existing drug for an original indication for a new indication. Here we have a table of 6 successful drugs, which were then from original indication repurposed also for a new indication. Why is this important? Well, there are high attrition rates, costs, and of course invested times, which are related to new drug discovery and development. And especially in the case of rare diseases, there is not enough pharmaceutical, let's say, incentive to go into them because the profits are just too small. But to repurpose a drug that is already existing on the market for a new rare disease, this is something that is totally feasible because these are de-risk compounds. Yeah? They are already approved by the regulators for the use in human implications. So this results in lower development costs and, of course, shorter development timelines. Now, drug repurposing is a complex process consisting of very different processes. But here you can see just the blue part of the circle represent the computational tools, while the, let's say, green part of the circle, the experimental procedures. So you see that 3/4 roughly, of drug repurposing are based on computational tools. And that's why computer chemistry, computer-aided drug design here really plays an immense role. So I will now briefly introduce two practical applications of our novel inverse docking procedure. First is the discovery of novel potential human targets of an already known molecule. So here in green on the left side, we have molecule resveratrol. It's a polyphenol from red grapes, red wine. It's supposed to be responsible for the French paradox that although they exist, they intake relatively fatty food in terms of cheese and liver pate, they have very little cardiac problems based on that diet. And this is partially attributed to the glass or 2 or 3 of red wine that French usually have during their lunch or dinner. So, but the mechanism of its action is relatively unknown. Why? Because polyphenols have many different OH groups, and consequently they represent so-called promiscuous binders. They don't bind just a single protein target, but various different protein targets. And this is very difficult for the experimentalists. And we want to shorten the list of possible protein interacting partners by using computational tools. So what do? We use the CanDoc molecular docking program, and we use it on the entire protein database from the protein data bank. That is this over 300,000 protein structures that are there already stored. Of course, this is generally an unsolvable problem because protein surfaces are large, and if we would have to scan the entire surface for docking, there are no computers strong enough on our planet to really do it. So we need to be smart. So we first detect binding sites of these proteins. And this we do with the help of the PROBIS algorithm, which is based on the evolutionary conservation of amino acids that have a specific role, like in the binding sites. So proteins outside the binding sites that have no function, they are more prone to mutagenesis, to change during time, whereas the important parts of the protein for the interactions, they remain relatively intact, so they have like evolutionary conserved amino acid residues there. By the inverse docking procedure, we get to target proteins, and of course, it's very good to see that if some of those target proteins that we identify already have the resveratrol structure in their crystal structure, and then we can compare the mode of binding of the two molecules, the one that is docked and the one that is pre-existent in the molecular structure. You see, they overlay really good, they really well, so we are satisfied with the results. What do we get? We get a table of the most prominent protein structure targets of our resveratrol. And here you can see where is denoted yes. For some of them, there is already a known experimental correlation with resveratrol, that the inhibition constant and that the IC50 were measured. For others, this still has to be performed, but we narrowed the scope of proteins that experimentalists have to examine by this computational tool from an impossible number of 300,000 to a table of 10 most likely protein targets. Another application is to study drug side effects. So here we have an example of two glitazone molecules which are used in the treatment of diabetes mellitus type 2. One of them is rosiglitazone, which is still on the market and is called Avandia, while the other one, troglitazone, was removed from the market. Why? Because of increased incidence of drug-induced hepatitis. And now, if we look at the most prominent protein targets of both molecules, we can see that they are really very much different. There is not much overlap between their most prominent protein targets. And really, we see that on the left, we have important inhibition of the cytochrome P450-2C8, which is in the liver and could cause the metabolic problems in the liver, whereas on the other side, for rosiglitazone, the most important target is PPR gamma. Again, you see redocking was very successful. So with this inverse molecular docking approach, we can really find the mechanistic basis of adverse side effects. But we, of course, want to go further into help to mitigate these effects or even to eliminate them. But, of course, what I owe you the most is how inverse docking applications can serve drug repurposing. So here I have 4 molecular fingerprints. 3 of these molecules, the ivermectin B1A, ivermectin B1B, and selemactin, they are all similar molecules from the macrolide family. And then there's a totally different molecule, darolutamide. And we can see that their molecular fingerprints are vastly different. The 3 macrolides are very similar in the nature, and the darolutamide very different from the 3. So what do we use for our filters? What is the x-axis of this graph? On the x-axis, we have different protein structures. So they are like molecular filters. So for every protein structure, we see how well this compound would bind to it. So we really get sort of a functional molecular zoo to get the similar and dissimilar compounds based on their fingerprints. Why is this important? Well, I mean, it's very difficult, even for experimentalists, to measure the interaction between a given molecule and, for example, 20 protein candidates. But once you have the fingerprints and you establish the similarity between molecules, not based on graphs or molecular properties, but really on their interaction with different proteins, then, of course, you can really speed up the experimental procedures. So here, I will give you a large heat map of 16 existing FDA-approved drugs, which were inversely docked into 142 different viral proteins. Why did we do this? Because we wanted to make the drug repurposing study for the SARS-CoV-2 virus for the COVID-19. Again, on top you see ivermectin, selamectin, very similar fingerprints. Then chloroquine, hydroxychloroquine. You probably remember there was a big fame if these two molecules could help in the disease. They have a similar, let's say, fingerprint, but not the one that would be a desired one. So for the SARS-CoV-2 main protease, we then successfully obtained 3 candidates from already existing drugs. One is olodaterol, the other one is xantinol, and the last one penbutolol, which are now in subsequent experimental studies. But what I want to say that this is a general process that could be fitted for any possible disease and any possible targets, and is especially valuable for the drug repurposing of rare diseases where the pharma companies don't have enough incentive, enough interest to really build new molecules, but maybe some of the existing drugs could already help in alleviating the related disease symptoms. With this, I would like to thank our computational resources in the form of High Performance Center Computing River in Maribor. We are very proud of it. It's one of the 50 strongest supercomputers worldwide, which is for Slovenia quite an achievement. And of course, I would like to thank you for your attention.
Thank you. Very nice talk. So did you find many difference between natural compounds and drugs? Are natural compounds more sticky or stick to many proteins, or what do you think? What's your view?
Yes, generally natural compounds are less selective. We could say that they are promiscuous. They interact with many different protein targets, whereas synthetic drugs tend to interact more specifically with the desired target for which they were actually experimentally designed and synthesized. So generally, the general rule would be for synthetic drugs, it is lower variability of targets and stronger binding. But for natural compounds, we would have a wider, more promiscuous amount of targets, but generally a weaker binding. This is the general finding that we get from our experiments. Thank you.
Thank you, Professor Bren.